Hi everyone, Mrs. Belo here. I'm just continuing from where I left off. So, again, in this problem here, you noticed your neighbor was, their garden was way better than yours, and you questioned why. So, your observation and then your question. And so, your hypothesis is if I use Miracle-Gro, then my garden will produce more produce, grow taller plants, etc. So the independent variable, what you are testing in this experiment is the Miracle-Gro, right? That is what you are testing. So if you are testing this on your garden, so you would probably have two gardens. One garden would be the control group, and that would be the group that does not get what you're testing, which is the Miracle-Gro fertilizer. So that would just be regular old, what you had before, your soil, nothing special about it. Versus the experimental group would be the group or the garden that is getting the Miracle-Gro, what it is you're trying to test. And then the dependent variable, that could be a lot of things that you could measure. You could measure to see if this Miracle-Gro is working. You can measure your plant's height with like a ruler. You can measure the number of plants that are growing. You can measure how much produce. Maybe you have a tomato plant. You're counting how many tomatoes it produces, okay? That could be collected over a certain amount of time, days, weeks, months, whatever. And then some things that you will have to keep the same throughout the experiment or control for would be things like water, soil, sunlight, maybe bugs, keeping bugs out or animals out, because you want to make sure the only thing influencing your results, influencing your experiment, is the independent variable, which in this case is the Miracle-Gro. All right, next, relationships. This is something that comes up a little bit more in chemistry, but I do like to mention it here. So when you graph two variables or two factors, whatever, on a graph, they could have hopefully one of two relationships. One can be a direct relationship. So this just means as one variable increases, the other increases as well. As one variable decreases, so does the other. And on a graph, that looks something like this. So for example, maybe even in that experiment with the fertilizer, maybe you even within that experiment, maybe you vary the amount of fertilizer. So maybe you're finding if you use more fertilizer, then you're seeing an increase in plant growth. That would be an example of a direct relationship, okay? Versus an inverse relationship is the opposite of that. So as one variable goes up, the other goes down. So maybe, going back to the science grade example, maybe like the more you play games or are distracted by your phone, so the more that you do that, the more time you spend doing those things, maybe on social media, maybe the lower your grade gets, or maybe your grade decreases. That would be an example of we don't say indirect, we don't want to say that, we say an inverse relationship. So those are just two types of relationships that you might see come up, inverse, direct. You might see those terms come up. Those are what they mean. All right, and then when you're collecting your data, okay, so after you've done the experiment, you've collected your data, your experiment should be without bias and repeated. So a lot of times scientists, it's not like they do one experiment and they're like, haha, I'm right. It doesn't work like that. Oftentimes experiments have to be repeated either by the same scientist or by other scientists. So oftentimes your experiment, it's something that has to be able to be repeated. And then also it should be completed without bias, meaning there was nothing else that you were doing to influence the results to have a favorable outcome. Especially like with the fertilizer, like you really wanted the fertilizer to work, okay? And so maybe you, you did something else to the garden, and that would be bias, okay? So you want to make sure that there is no bias. You are being very objective in your experiment and not subjective. And even that includes reporting your data. So data can be communicated with graphs, charts, or tables. And usually it has to be in some organized fashion, maybe even a pie chart, a bar graph, a table that's organized. Raw data isn't always the easiest to interpret or look at. Usually that data has to be put into some organized format. Sampling size is also important. Which is better, a large or a small sample size? You definitely want a large sample size. Again, this can lead to bias, right? If you have just a small, let's say, group of people you're experimenting with, or maybe you're doing a survey, I don't know. You don't want to have a small sample size because then you're not getting an accurate representation, okay? So a large group is going to be more representative. Okay? You don't, because like what if you sample a small group of people and you're asking them, you know, if they like, I don't know, apples or oranges, and like almost all of them say oranges and one person says apples, and you only ask 10 people. Well, you might get different results if you ask like 100 or 1,000 people, okay? That's really important to keep in mind, especially if you are ever looking or reading about a study that was done. Find out how many people were involved in it or how many people were surveyed, because that is very important. Data can be collected as numerical data. This is usually the most common one we see. And we call this quantitative data. So this data is numerical, right? And we use this a lot, especially in science, like how much, okay, you measure in centimeters, you weigh something in grams, you take volume in liters, you know, how many people said they like this, okay? That's all numerical data. And that's the most common data that we use. So for example, a plant that grew 6 inches, 6 inches would be the quantitative data. Versus if we're talking about descriptive data, that is qualitative. So maybe in that experiment with the plants, maybe you didn't just measure the height of the plants, maybe you also noted the color of the leaves. Maybe some plants had greener leaves than others, maybe some were more yellow. That's something that you can't really record necessarily in numbers, it's more descriptive data, and that can be also really important and relevant as well. Here's some other examples. So again, I always think quantitative. I always remember it. I think quantity. I think like how much. Okay. So I think quantity. I think how much. Versus qualitative. I think of quality. So quantitative. I think how much. So numbers. Weight, shoe size, scores on an exam, that's all numerical. That is asking about how much. Versus descriptive data, qualitative data is like color, maybe smell, names. That's all quality related things or descriptive. Okay, so once you've asked a question, you've come up with a hypothesis, you designed an experiment, you tested something more than once, okay? Other people have tested it, and you've analyzed your results, both qualitative and quantitative data. Usually that's like the best is to report both, but either one can be fine. And now you can talk about your conclusions. What can you conclude from this experiment? Okay, was your hypothesis supported or rejected? And if you do an experiment enough, you support a hypothesis enough, it can lead to constructing a theory. Now, oftentimes this is used in everyday language. Oh, that's just a theory. But in the science context, in the science world, a theory is really important. A theory, as you can see here in this diagram, is basically a bunch of hypotheses and testable explanations, things that have been supported by a large body of evidence and many hypotheses. This is something that is thought to be true because it has been tested and supported so many times. For example, there is the theory of evolution by natural selection, so explaining how species change over time. There's quantum theory. It's a little bit outside my realm of expertise. That's more physics. Same thing, general relativity. That's Albert Einstein's theory of relativity. There's cell theory. We'll talk about that later in our cells unit. Atomic theory. These are things that have been supported. Newton's law of motion, what is in motion stays in motion unless stopped by another object, right? Or that an object in its current state, rest or in motion, can stay that way unless it's affected by another force. So their theories are things that have a lot of supporting evidence behind them. And when it comes to publishing work, so scientists, I had to do this for my master's degree. Scientists, when they publish work, they communicate that with each other. And a lot of times, if you go into a database, like through Illinois State University, I have access to a database through them. You want to make sure that if you are looking at studies, you're looking at ones that have been peer reviewed. And again, so peer review, so allows for peer review. Basically, this is when someone's work has been authenticated or checked by someone else. So one scientist has done an experiment, and got certain results. Another scientist did the same experiment and they got similar results. That's supporting the original scientist hypothesis. Because again, it's possible scientists could do an experiment and it could be biased. So it's important that other people are testing the hypothesis and doing the same experiment as well. It helps us to determine, of course, where money ultimately can go and to avoid conflict, to solve problems, because we don't want to be putting information out there that is not true.